So I'm Patricia Inácio, Bionews Service Correspondent for Friedrich Ataxia News. Dr. Meyer, thank you so much for being part of Friedrich's Ataxia News official coverage of the International Ataxia Research Conference here in Pisa, Italy, and to agreeing to being filmed and recorded for this interview. So our first question is, Reata is developing omaveloxolone as a treatment for Friedrich's ataxia, but the company is also testing the drug in mitochondrial myopathies and melanoma. Are the underlying molecular processes of these three disorders similar, and is omaveloxolone's mechanism of action largely the same in these three medical conditions?
Sure. Thank you for having me. Our drug, omaveloxolone, as it's now known, or OMAV, was formerly known as RTA408, and within the cell it acts by inducing an important protein called NRF2. And the primary effect of induction of NRF2 is restoration of mitochondrial energy production. It also dampens down inflammation within the cell. And so the underlying mechanism is the same, but the context, of course, is different across the diseases. And the activity in Friedreich's ataxia is very similar and applicable to the patients who also have mitochondrial disease and mitochondrial myopathies. And the primary effect is restoration of energy production within the cells that then translates to improved organ function in the muscles, in the brain, and elsewhere.
So omaveloxolone is being assessed in the ongoing phase 2 MOXIE trial. Later today, Dr. David Lynch will present results from part 1 of this trial. How well did the results meet Reata's expectations?
The results were very encouraging and met all of our expectations. The trial that was conducted was a phase 2 trial. It's called MOXIE Part 1. It's the largest phase 2 trial conducted in Friedreich's ataxia. And so it enrolled 69 patients across 7 dose levels. It was placebo-controlled. And importantly, we assessed many different parameters of interest. So we were able to show in patients with Friedreich's ataxia that OMAV could induce NRF2. And so we monitored several biological targets, and so the drug hits its primary biological target. We're also able to show that the drug improves markers of mitochondrial function. We also demonstrated that the drug has activity on many important neurological parameters, such as the modified FARS, and we identified a dose that had the most optimal activity. The drug was also well tolerated with no discontinuations at the optimal dose. And so that then allowed us to demonstrate that the drug was active, appeared to have meaningful clinical activity, and so we're now initiating the registrational portion of the trial with the optimal dose in 100 patients globally.
So concerning to some of the things that you told us now, so results from the MOXIE trial show that OMAV significantly improve neural function in FA patients. An overall improvement in mFARS of 3.8 compared to baseline at the 160 milligrams dose. Was this improvement clinically meaningful in patients? Which neural capacities were improved?
Yes, and so as the FARA, as well as the international community, has demonstrated, the average change in the modified FARS score annually is about 1 to 2 mFARS points. And so at the optimal dose in the phase 2 portion of the trial, we demonstrated a placebo-corrected change of about 2.2 points, which would equal about 1 year of progression. And so patients were able to recover what they typically would lose in 1 year in only a 12-week trial. We did note that in a subset of patients who did not have a foot deformity called pes cavus, that their activity was greater, and the placebo-corrected change was about 4.4 points, which represents about 2 years worth of progression. And so we believe that those changes are very clinically meaningful.
The MOXIE trial is enrolling people between the ages of 16 to 40. Is Reata planning to eventually test OMAV in younger patients?
Yes, ultimately, we will do additional clinical testing for the purpose of the MOXIE trial. We try to limit the patients so that they're very similar and homogeneous, so we can reduce the noise in the trial and hopefully optimize it for success.
Enrolled patients must have a modified Friedreich's ataxia rating scale, mFARS, of at least 20, but not greater than 80. Knowing that the higher the score, the worse is the patient's disability, is Reata planning to test OMAV in patients with more advanced disability?
Yes, ultimately, we would like to test the drug in other patients who do not qualify for this trial. But once again, we need to try to maximize the chances of success in this initial trial so that we can hopefully get the first approved drug to these patients. And then obviously we can do additional trials in a broader set of patients.
Findings in Part 1 seem to imply a lesser benefit in treated patients with musculoskeletal foot deformities, pes cavus. These patients are generally in more advanced disease stages. In this finding, possibly pointed to OMAV being a better treatment for FA patients still in early or middle disease stages, and is testing this possibly the reason that a limited number of patients, up to a maximum of 20% of enrollment, we believe, in Part 2 can be pes cavus patients.
Yes, so we importantly noted in the phase 2 results that patients who had pes cavus showed a smaller effect on the drug than patients who did not. We think the drug works similarly across these patients. It's just harder to see the changes in patients with pes cavus. We noted that in the modified FARS endpoint that most of the improvements came from upright stability and lower limb coordination, so the patient's ability to walk and stand. For patients that have pes cavus, which is associated with a high arch foot, they have a structural deficit in their foot that makes it difficult for them to stand completely apart from their ataxia. And so for that reason, we think it was hard to see the changes in those patients, and so we are limiting those patients in the trial. Importantly, they are being included, and so if the trial is successful, hopefully the drug will be available for those patients. But once again, we need to try to maximize the chances of success of this trial, and so we're limiting those patients.
Congratulations on the therapy receiving an orphan drug designation from the FDA in June. If the MOXIE trial shows robust clinical evidence that OMAV is an effective and safe therapy for FA, what next steps is Reata considering in the drug's development program?
Thank you very much. We're excited to receive the orphan drug designation for omaveloxolone for Friedreich's ataxia. Importantly, the Part 2 of MOXIE is a registrational trial, and so it's like a Phase 3 trial in that if this study is successful, we believe it would support registration not only in the U.S., but globally. And so we need to enroll the trial, obviously follow patients for a total of 48 weeks, and then analyze the data. And if the data show appropriate efficacy and safety, then we would plan to start working with regulators globally to hopefully submit the drug for approval so that it could be the first approved treatment for Friedreich's ataxia.
Is there anything else you'd like to discuss or further clarify?
I'd just like to reiterate that the second part of the MOXIE trial is registrational, meaning that it will support approval if it's successful. It's the most advanced program globally right now in the Friedreich's ataxia in development. And so we're actively working with the investigators, FARA, other patient advocacy groups to help facilitate enrollment in this important trial.